Tonight we begin with a question that has captivated humanity for centuries. What secrets lie buried in stone? What stories do these ancient remnants whisper about worlds that vanished long before our first breath? In the quiet corners of museums and the dusty layers of sedimentary rock, fossils wait patiently to reveal the most extraordinary tale ever told, the true history of life on Earth. Before you settle in for this gentle journey through deep time, take a moment to find your most comfortable position. Perhaps dim the lights, let the day's concerns fade away, and allow yourself to drift into a state of peaceful curiosity. We're about to embark on a voyage that spans billions of years, following the evidence left behind by creatures that once walked, swam, and flew through ancient landscapes that no human eye has ever seen. What we'll discover together isn't speculation or fantasy. Every detail we explore tonight is grounded in solid evidence, carefully extracted from the fossil record by generations of dedicated scientists who've spent their lives reading the language of stone. These aren't just old bones and shells. They're time capsules, each one preserving a moment from Earth's vast biography, waiting to tell us exactly what prehistoric life was really like. Now, settle back and let your imagination wander as we begin this tranquil exploration of deep time, where every fossil holds a piece of the puzzle that is life's extraordinary journey. The story of fossils begins not with death, but with the remarkable circumstances that allow life to cheat time itself. In the natural world, death usually means complete recycling. Bacteria, scavengers, weather, and chemical processes work together to break down organic matter and return it to the ecosystem. But occasionally, under very specific conditions, something extraordinary happens. The normal processes of decay are interrupted, and instead of vanishing forever, an organism begins its transformation into stone. Picture a shallow lagoon 200 million years ago. The water is warm and still, rich with dissolved minerals. A small marine reptile, perhaps an early ichthyosaur, dies and sinks gently to the muddy bottom. Normally, this would be the end of its story. But the conditions here are special. The water lacks oxygen near the bottom, which means the bacteria that would ordinarily consume soft tissues cannot survive. Fine sediment, carried by gentle currents, begins to settle over the body like a soft blanket. This is the beginning of fossilization, a process so unlikely that paleontologists estimate only a tiny fraction of all organisms that have ever lived have left any trace in the fossil record. The burial must happen quickly before scavengers can scatter the bones or currents can wash them away. The sediment must be fine enough to capture delicate details, yet contain the right chemistry to preserve hard tissues over millions of years. As the layers of sediment accumulate above our ancient reptile, the weight begins to compress the surrounding material. Groundwater, rich with dissolved minerals, percolates through the sediment layers. Slowly, grain by grain, molecule by molecule, minerals begin to replace the original bone material. Calcium phosphate in the bones may be replaced by silica, iron compounds, or other minerals present in the surrounding environment. This process, called permineralization, can take thousands of years to complete. The original shape and even microscopic details of the bone are preserved as the minerals crystallize within the bone's porous structure. In some cases, the preservation is so exquisite that scientists can examine individual cells under powerful microscopes. The fossil becomes heavier and denser than the original bone, transformed into a stone replica that can survive for hundreds of millions of years. But body fossils are just one type of evidence that prehistoric life has left behind. Perhaps even more fascinating are trace fossils, the preserved signs of ancient behavior. These include footprints pressed into ancient mud, burrows dug by long extinct creatures, bite marks on bones, and even fossilized dung called coprolites. Unlike body fossils, which tell us what an organism looked like, trace fossils reveal what these ancient creatures actually did. Consider the famous dinosaur trackways found in places like the Connecticut River Valley or the Paluxy River in Texas. These aren't random footprints, but carefully preserved sequences of steps that allow paleontologists to calculate walking speeds, determine whether dinosaurs moved in herds, and even identify moments of interaction between different species. Some trackways show clear evidence of predator and prey relationships, with the footprints of a large carnivorous dinosaur following those of a smaller herbivore. The process of preserving footprints requires conditions even more specific than those needed for body fossils. The sediment must be exactly the right consistency, firm enough to hold the shape of the foot, but soft enough to receive a clear impression. The print must then be covered quickly by different sediment before wind, rain, or other animals can destroy it. When conditions align perfectly, these trace fossils can preserve behavioral details that no amount of skeletal evidence could reveal. In rare and precious circumstances, even soft tissues can be preserved. The famous Burgess Shale in British Columbia, Canada, dating back over 500 million years, contains fossils of creatures that were entirely soft-bodied. These exceptional preservation sites, called Lagerstätten by paleontologists, form under very special conditions. Often, they require environments with no oxygen, which prevents bacterial decay, combined with fine sediments that can capture microscopic details. The Solnhofen limestone in Germany provides another window into exceptional preservation. Here, the famous specimen of Archaeopteryx was discovered, complete with detailed impressions of its feathers. The limestone formed in a tropical lagoon environment where the water was so salty and lacking in oxygen that virtually nothing could survive to disturb the settling organisms. Bodies sank into the fine carbonate mud and were preserved in stunning detail. These sites reveal aspects of prehistoric life that would be completely unknown from ordinary fossils. Soft-bodied jellyfish, worms, and other creatures that normally leave no trace are preserved alongside more familiar hard-shelled organisms. The diversity of life revealed at these special sites suggests that the normal fossil record, dominated by shells, bones, and teeth, represents only a small fraction of the organisms that actually lived in ancient times. Understanding how fossils form is crucial for interpreting what they tell us about prehistoric life. Every fossil comes with built-in limitations. The fossil record is biased toward organisms with hard parts, toward creatures that lived in environments where burial and preservation were likely, and toward the most recent chapters of Earth's history. Marine organisms are much more likely to be preserved than terrestrial ones because ocean floors and lake beds provide the steady sedimentation necessary for fossilization. This preservation bias means that our view of ancient ecosystems is necessarily incomplete. Forest ecosystems, which are dominated by soft-bodied insects, spiders, and other arthropods, are poorly represented in the fossil record compared to marine ecosystems with their abundant shell-bearing creatures. Similarly, juvenile animals and small organisms are less likely to be preserved than large adults, skewing our understanding of ancient population structures. Yet despite these limitations, the fossil record provides an extraordinarily rich source of information about life's history. Modern paleontological techniques can extract far more information from fossils than early collectors ever imagined. Chemical analysis can reveal what ancient animals ate by examining isotope ratios in their bones and teeth. Microscopic examination can reveal details about growth rates, seasonal changes in diet, and even stress markers that indicate environmental pressures. The study of how organisms become fossils, called taphonomy, has become a sophisticated science in its own right. By understanding the processes that affect remains between death and final burial, paleontologists can better interpret what fossils represent and avoid drawing incorrect conclusions about ancient life and environments. Perhaps most remarkably, recent advances have pushed the boundaries of what can be preserved and studied. Scientists have recovered ancient proteins from fossils millions of years old, providing direct chemical evidence of evolutionary relationships. Even more extraordinary, researchers have extracted and sequenced RNA from woolly mammoth specimens, frozen for nearly 40,000 years, offering unprecedented insights into the molecular biology of extinct organisms. As we examine these various modes of preservation, we begin to appreciate that fossils are not simply old bones and shells, but rather the products of complex physical and chemical processes that occasionally conspire to preserve moments from the deep past. Each fossil represents a minor miracle of preservation, a convergence of circumstances so unlikely that most organisms leave no permanent record of their existence. The formation of fossils requires not just the right environmental conditions, but also the passage of vast amounts of time. The transformation from organic remains to stone fossil occurs gradually, as minerals slowly replace original tissues and sediments compact into solid rock. This process connects us directly to the deep history of our planet, allowing us to touch stones that were once living creatures swimming in ancient seas or walking across primordial landscapes. When we hold a fossil in our hands, we're holding evidence of life that existed in worlds almost unimaginably different from our own. The continents were in different positions, the climate was often warmer or cooler than today, and the mix of gases in the atmosphere bore little resemblance to the air we breathe. Yet life flourished in these ancient environments, leaving behind traces that allow us to reconstruct their stories with remarkable precision. The rarity of fossilization makes every specimen precious. Paleontologists estimate that significantly less than 1% of all species that have ever lived are known from fossils. This means that for every dinosaur species we've discovered, hundreds or thousands of others lived and died without leaving any trace we've yet found. The fossil record is like a vast library where most of the books have been lost, and we're left to reconstruct the complete story from the scattered pages that survive. Despite these limitations, the pages we do have tell an extraordinary story. Fossils provide our most direct evidence for evolution, documenting the gradual changes that have transformed life over billions of years. They reveal the timing of major evolutionary innovations, from the first appearance of hard shells to the development of flight. They show us how life has responded to environmental changes, including climate shifts and mass extinction events that periodically reshaped the biological world. The process of reading this stone library requires patience, careful observation, and increasingly sophisticated analytical techniques. Modern paleontologists combine traditional fieldwork with advanced laboratory methods, using everything from electron microscopy to isotope geochemistry to extract maximum information from fossil specimens. Each new analytical technique opens new windows into the past, revealing details about ancient life that previous generations of scientists could never have imagined. As our understanding of fossilization processes has improved, so has our ability to find and interpret fossils. Paleontologists now know what kinds of environments are most likely to preserve remains, allowing them to target their searches more effectively. They understand how different burial conditions affect preservation, helping them interpret what fossil assemblages represent about ancient ecosystems and environments. The study of exceptional preservation sites has been particularly revealing. These windows into ancient soft tissue preservation show us that the diversity of prehistoric life was far greater than what ordinary fossil sites suggest. They reveal entire categories of organisms that would be completely unknown if we relied only on typical preservation of hard parts. Perhaps most importantly, understanding fossilization helps us appreciate both the power and the limitations of the fossil record. While we can never recover a complete picture of any ancient ecosystem, the evidence we do have provides remarkable insights into how life has changed through time and how ancient organisms lived, grew, reproduced, and interacted with their environments. This foundation in understanding how fossils form and what they represent prepares us for a deeper exploration of what these ancient remains actually tell us about prehistoric life. Armed with knowledge of preservation processes and their limitations, we can begin to read the stories written in stone with greater confidence and precision. The first step in reading the fossil record is learning to see time differently. When we examine a fossil, we're not just looking at an ancient organism, we're glimpsing a moment preserved from Earth's 4.5 billion year history. But understanding what that moment represents requires us to place it accurately in time, and this process of dating fossils has become one of the most sophisticated aspects of modern paleontology. The principles of relative dating were established long before scientists understood radioactive decay. In the late 18th century, geologists realized that rock layers, or strata, are deposited in sequence, with older layers beneath younger ones. This principle of superposition provides a basic framework for understanding the relative ages of fossils. A trilobite found in a layer beneath one containing early fish must be older than those fish, even if we don't know the exact ages of either. But relative dating only tells us which fossils are older or younger than others. To understand the true scope of geological time and the pace of evolutionary change, paleontologists need absolute dates. The discovery of radioactive decay in the early 20th century provided the key to unlocking these precise ages. Certain elements naturally break down into other elements at predictable rates, creating atomic clocks that tick away steadily over millions and billions of years. Carbon-14. Dating, perhaps the most familiar radiometric technique, works well for relatively recent organic materials. When organisms are alive, they constantly exchange carbon with the environment through breathing, eating, and photosynthesis. This includes a tiny amount of radioactive carbon-14, which maintains a constant ratio with stable carbon-12. When an organism dies, the exchange stops, and the carbon-14 begins to decay with a half-life of about 5,700 years. By measuring how much carbon-14 remains in a fossil compared to living organisms, scientists can determine when the organism died. This technique is invaluable for studying late Pleistocene fossils, including many human ancestors and the creatures that lived alongside early humans during the last ice age. However, carbon-14 decays relatively quickly on geological timescales, making it useful only for materials less than about 50,000 years old. For older fossils, paleontologists turn to longer-lived radioactive elements. Potassium-argon dating utilizes the decay of potassium-40 into argon-40, a process with a half-life of over 1 billion years. This makes it ideal for dating very ancient rocks and the fossils they contain. When volcanic rocks form, they trap radioactive potassium but contain no argon gas. As time passes, the potassium decays into argon, which remains trapped in the rock crystals. By measuring the ratio of potassium to argon, scientists can determine when the rock formed. This technique is particularly valuable for dating fossil sites in areas with volcanic activity. The famous hominin fossils from East Africa, for instance, are often found in sediments sandwiched between layers of volcanic ash. By dating these ash layers above and below the fossil-bearing sediments, paleontologists can bracket the age of the fossils with remarkable precision. Uranium-lead dating provides another tool for examining the most ancient chapters of Earth's history. Uranium-238 decays through a complex chain of intermediate elements to eventually become lead-206, with a half-life of over 4 billion years. This technique has been crucial for dating some of the oldest known fossils, including microscopic evidence of early life that formed more than 3 billion years ago. The integration of multiple dating techniques often provides the most reliable results. When different methods applied to the same sample yield similar ages, scientists can have confidence in their conclusions. When results disagree, it often points to interesting geological processes or contamination that requires further investigation. Beyond radiometric dating, paleontologists use a variety of other techniques to establish temporal frameworks. Magnetic stratigraphy takes advantage of the fact that Earth's magnetic field has reversed many times throughout geological history. When rocks form, they record the orientation of the magnetic field at that time. By mapping these magnetic reversals in rock sequences, scientists can correlate rock layers from different locations and establish precise temporal relationships. Biostratigraphy uses the evolutionary changes in fossils themselves as dating tools. Certain groups of organisms evolved rapidly and spread widely, making them excellent index fossils for dating rock layers. Trilobites, ammonites, and various microscopic marine organisms provide particularly useful biostratigraphic markers because they evolved quickly and are found in marine sediments worldwide. The combination of these various dating methods has revealed the extraordinary length of geological time and allowed paleontologists to construct detailed timelines of evolutionary history. We now know that complex life appeared in the oceans over 500 million years ago, that dinosaurs dominated terrestrial ecosystems for more than 160 million years, and that our own species has existed for only about 300,000 years. This temporal precision transforms our understanding of evolutionary processes. We can see that some changes happened gradually over millions of years, while others occurred rapidly during periods of environmental crisis. The fossil record reveals that evolution is not a steady, uniform process, but rather one characterized by long periods of relative stability, punctuated by episodes of rapid change. Understanding geological time also helps us appreciate the rarity and preciousness of individual fossils. A single specimen might represent the only known example of a species that lived for millions of years. Each fossil is an ambassador from a specific moment in Earth's vast history, carrying information about conditions that existed at that precise time and place. The precision of modern dating techniques has also revealed unexpected complexity in the fossil record. What once appeared to be simple evolutionary sequences often turn out to be more complicated stories of branching lineages, convergent evolution, and extinction events. The ability to date fossils precisely has shown that evolution is far more dynamic and intricate than early paleontologists imagined. As we move deeper into examining what fossils tell us about prehistoric life, this temporal framework provides the essential scaffolding for understanding evolutionary relationships and environmental changes. Knowing when organisms lived allows us to reconstruct ancient ecosystems, track the movements of continents, and understand how life has responded to changing environmental conditions throughout Earth's history. The story that emerges from precisely dated fossils is one of constant change over unimaginable spans of time. Continents have drifted across the globe, ocean levels have risen and fallen by hundreds of feet, and climates have swung between ice ages and greenhouse periods. Through it all, life has persisted, adapted, and diversified, leaving behind the fossil evidence that allows us to reconstruct this remarkable history. With this temporal foundation established, we can begin to explore the specific insights that different types of fossils provide about how prehistoric organisms lived, moved, fed, and interacted with their environments. The stories written in stone become far more meaningful when we understand not just what these ancient creatures looked like, but when they lived, and how their world differed from our own. The anatomy revealed by body fossils provides our most direct evidence of how extinct organisms were constructed and how they functioned in their ancient environments. Every bone, shell, and tooth carries information about the organism's lifestyle, from the microscopic details of bone structure to the overall proportions that determined how efficiently an animal could move, feed, or defend itself. Consider the remarkable preservation found in a typical dinosaur skeleton. The shape and size of leg bones immediately reveal information about locomotion. Long, slender bones suggest a fast-running animal, while robust, heavily built bones indicate a slower, more powerful creature. The angle of joints shows the range of motion available to ancient limbs, allowing paleontologists to reconstruct gaits and estimate running speeds. The skull provides perhaps the richest source of anatomical information. Tooth shape and wear patterns reveal diet with remarkable precision. Sharp, pointed teeth indicate a carnivorous lifestyle, while flat, grinding surfaces suggest plant-eating habits. The size and shape of eye sockets indicate whether an animal was active during the day or night, while inner ear structures preserved in fossilized skulls can reveal information about balance and hearing capabilities. Recent advances in CT scanning technology have revolutionized the study of fossil anatomy. Scientists can now examine the internal structures of fossil skulls without damaging precious specimens. These techniques have revealed brain shapes in extinct animals, providing insights into intelligence and sensory capabilities. The famous Tyrannosaurus rex specimen known as Sue yielded surprising information about brain structure when CT scanned, showing that these massive predators had relatively sophisticated sensory processing abilities. The preservation of microscopic details in fossils often provides the most fascinating insights. Bone histology, the study of bone tissue structure, reveals how fast ancient animals grew and whether they were warm-blooded or cold-blooded. The presence of growth rings in bones, similar to tree rings, shows seasonal variations in growth rate and can even indicate the age at which an animal died. Some fossils preserve evidence of injuries and diseases that affected ancient organisms. Broken bones that healed improperly, arthritis in joints, and even evidence of parasitic infections provide glimpses into the challenges faced by individual animals millions of years ago. These pathological specimens remind us that prehistoric life involved the same struggles with injury, disease, and aging that affect modern organisms. The study of fossil anatomy has revealed remarkable convergent evolution, where unrelated organisms developed similar body plans in response to similar environmental pressures. The streamlined bodies of extinct marine reptiles like ichthyosaurs closely resemble those of modern dolphins and sharks, despite evolving from completely different ancestral groups. This convergence demonstrates the power of natural selection to produce optimal solutions to environmental challenges. Exceptional preservation sometimes captures anatomical details that would normally decay immediately after death. The famous feathered dinosaur fossils from China preserve not only the presence of feathers, but also their detailed structure and arrangement. These discoveries have revolutionized our understanding of dinosaur biology, showing that many species were covered with complex plumage that served functions ranging from insulation to display. The preservation of soft tissues, while extremely rare, provides unparalleled insights into the biology of extinct organisms. Fossilized stomach contents reveal the last meals of ancient animals, providing direct evidence of predator-prey relationships and dietary preferences. Preserved eggs and embryos show developmental stages and parental care behaviors that would be impossible to infer from adult skeletons alone. The anatomical evidence preserved in fossils often challenges our assumptions about how extinct organisms lived. Many dinosaurs once thought to be sluggish. Tail-dragging reptiles are now understood to have been active, agile animals with complex behaviors and sophisticated sensory capabilities. The discovery of evidence for warm-bloodedness in dinosaurs, derived from bone histology and other anatomical features, has fundamentally changed our understanding of their biology and ecology. Fossil anatomy also reveals the evolutionary transitions between major groups of organisms. The famous specimen of Archaeopteryx preserves both reptilian features like teeth and a long bony tail, and avian features including feathers and wishbone. This anatomical mosaic provides direct evidence for the evolutionary transition from dinosaurs to birds, showing how major evolutionary innovations arose through the modification of existing structures. The study of plant anatomy and fossils reveals similar insights about ancient terrestrial ecosystems. Fossilized wood structure shows how ancient trees responded to seasonal changes and environmental stresses. Leaf anatomy provides information about ancient atmospheric conditions, as plants adjust their leaf structure in response to carbon dioxide levels and humidity. The preservation of reproductive structures in both plants and animals offers glimpses into the evolution of sexual reproduction and parental care. Fossil flowers show the coevolution of plants and their pollinators, while fossilized eggs and nests reveal the development of increasingly sophisticated reproductive strategies in vertebrates. Perhaps most remarkably, recent discoveries have pushed the boundaries of anatomical preservation to the cellular level, Some fossils preserve actual cellular structures, allowing scientists to study the microscopic organization of organisms that died hundreds of millions of years ago. These discoveries blur the line between paleontology and cell biology, opening new avenues for understanding the fundamental processes of life. The anatomical information preserved in fossils provides the foundation for understanding all other aspects of prehistoric life. Body structure determines how organisms moved, what they could eat, where they could live, and how they interacted with other species. This anatomical evidence, combined with environmental information from the surrounding rock layers, allows paleontologists to reconstruct ancient ecosystems with remarkable detail and accuracy. While body fossils tell us what ancient organisms looked like, trace fossils reveal what they actually did. These preserved signs of ancient behavior provide intimate glimpses into the daily lives of creatures that lived millions of years ago, showing us how they moved, fed, reproduced, and interacted with their environments in ways that no amount of skeletal evidence could reveal. Footprints represent perhaps the most evocative type of trace fossil. When we look at a dinosaur trackway, we're seeing the actual path taken by a living animal across an ancient landscape. These tracks capture moments of behavior with stunning immediacy, preserving not just the size and shape of feet, but also stride length, walking speed, and evidence of social interactions between different individuals. The famous trackways at Dinosaur Ridge in Colorado tell stories of daily life in a Cretaceous landscape. Here, the footprints of various dinosaur species intersect and overlap, showing how different animals used the same pathways at different times. Large sauropod tracks reveal the ponderous movement of massive long-necked dinosaurs, while smaller theropod tracks suggest the quick, efficient gait of predatory species. Careful analysis of trackway geometry allows paleontologists to calculate walking and running speeds with remarkable precision. The relationship between stride length, foot size, and estimated body weight provides a formula for determining how fast ancient animals moved. Some dinosaur trackways preserve evidence of running speeds exceeding 20 miles per hour, challenging earlier assumptions about the athleticism of these ancient creatures. Group trackways provide evidence for social behavior that would be impossible to infer from skeletal remains alone. Parallel trackways of similar-sized individuals suggest herd movement, while trackways showing adults and juveniles together provide evidence for family groups and parental care. The famous sauropod trackways from Texas show evidence of herd migration, with adults apparently surrounding juveniles for protection during long-distance travel. Predator-prey interactions occasionally become frozen in stone through trackway evidence. Sites in Texas and other locations preserve trackways where the footprints of large carnivorous dinosaurs follow those of potential prey species, creating fossilized chase scenes that played out over 100 million years ago. While we can't know the outcome of these ancient pursuits, the tracks provide dramatic evidence of the predator-prey relationships that shaped Mesozoic ecosystems. Swimming traces preserved in ancient lake and marine sediments reveal the aquatic behaviors of organisms that also lived on land. Some dinosaur trackways show the distinctive pattern of an animal swimming rather than walking, with only the tips of toes touching the sediment as the body was buoyed by water. These traces provide evidence for the swimming abilities of terrestrial dinosaurs and help reconstruct the margins of ancient water bodies. Burrows and other dwelling traces reveal how ancient organisms modified their environments for shelter, food storage, and reproduction. The complex burrow systems of ancient mammals, preserved in Paleocene and Eocene sediments, show sophisticated architectural behaviors that parallel those of modern social mammals. These underground networks often preserve evidence of multiple generations using the same burrow systems, suggesting complex social structures and territorial behaviors. Marine trace fossils include the feeding traces left by ancient organisms as they searched for food in seafloor sediments. These traces, called ichnofossils, often preserve intricate patterns that reveal feeding strategies, hunting techniques, and the organization of ancient benthic communities. Some feeding traces show clear evidence of learning and optimization, with later portions of the trace showing more efficient search patterns than earlier sections. Bite marks on fossil bones provide direct evidence of predator-prey interactions and scavenging behaviors. The spacing and depth of tooth marks can identify the species responsible for the damage, while the healing patterns around bite marks show whether animals survived attacks or were consumed after death. Some fossil bones preserve multiple sets of bite marks from different predator species, revealing the complex scavenging hierarchies of ancient ecosystems. Coprolites, or fossilized dung, provide remarkably intimate insights into the diets and digestive systems of ancient organisms. These trace fossils preserve direct evidence of what extinct animals actually ate, often including fragments of prey species, plant material, and parasites. The size and shape of coprolites can often be matched to specific producer species, while chemical analysis reveals detailed information about digestive efficiency and metabolic rates. The study of coprolites has revealed surprising dietary diversity in many extinct species. Some dinosaur coprolites contain evidence of both plant and animal material, suggesting more omnivorous diets than previously suspected. Others preserve evidence of seasonal dietary changes or specialized feeding behaviors that wouldn't be apparent from tooth structure alone. Nesting traces provide evidence for reproductive behaviors and parental care in extinct species. Dinosaur nesting sites preserve not just the eggs themselves, but also evidence of nest construction, egg arrangement, and brooding behaviors. Some sites show evidence of colonial nesting, with multiple individuals using the same area for reproduction over many seasons. The famous dinosaur nesting sites in Montana and other locations have revealed sophisticated parental care behaviors, including evidence that some species brought food to their young in the nest. The arrangement of eggshells and juvenile remains provides insights into hatching success, growth rates, and the duration of parental care in species that died out millions of years ago. Tool use traces, while rare, provide evidence for complex behaviors in extinct species. Some sites preserve evidence of early human ancestors using stones to crack nuts or process other foods, with characteristic damage patterns on both the tools and the food remains. These traces push back the earliest evidence of tool use and provide insights into the cognitive development of early hominins. Trace fossils often preserve behaviors in organisms that left no body fossils at all. Soft-bodied creatures that would otherwise be unknown from the fossil record sometimes leave distinctive traces that reveal their presence and activities in ancient ecosystems. These traces provide our only evidence for entire groups of organisms that played important ecological roles, but lacked preservable hard parts. The study of trace fossils has revealed that many behaviors we consider distinctly modern actually have very ancient origins. Evidence for complex social interactions, sophisticated feeding strategies, and advanced parental care appears much earlier in the fossil record than once thought. These discoveries remind us that intelligence and complex behavior are not recent innovations, but rather have deep evolutionary roots. Perhaps most importantly, trace fossils capture the dynamic aspects of ancient life that body fossils alone cannot reveal. They show us that prehistoric organisms were not static museum displays, but rather active, behaving creatures that moved, fed, reproduced, and interacted with their environments in complex ways. These behavioral fossils bring ancient ecosystems to life in ways that skeletal remains alone never could, providing our most direct connection to the daily experiences of creatures that lived in worlds vastly different from our own. The combination of body fossils and trace fossils provides a remarkably complete picture of ancient life, revealing not just what prehistoric organisms looked like, but how they lived, moved, and behaved in their ancient environments. Together, these different types of evidence allow paleontologists to reconstruct ancient ecosystems with a level of detail and accuracy that continues to surprise and delight scientists and the public alike. The public alike. The fossil record reveals far more than scattered bones and shells. It documents the intricate web of relationships that connected ancient organisms to each other and to their environments. When paleontologists examine fossil assemblages, they're looking at snapshots of entire ecosystems frozen in time, complete with predators and prey, plants and pollinators, and the complex interactions that sustained life millions of years ago. Understanding these ancient ecological relationships requires careful analysis of multiple lines of evidence. The size and abundance of different fossil species within a single rock layer can reveal the structure of ancient food webs. Large predators are typically rare compared to their prey, just as in modern ecosystems, and this pattern is clearly preserved in fossil assemblages. When paleontologists find numerous small herbivorous dinosaur fossils alongside fewer, larger carnivorous species, they can reconstruct the basic energy flow of these ancient communities. The chemistry preserved in fossil bones and teeth provides even more detailed information about ecological relationships. Isotope analysis can determine what ancient animals ate and where they obtained their food. Carbon isotopes in tooth enamel reveal whether herbivorous dinosaurs fed primarily on ferns and conifers or on the flowering plants that appeared later in their evolutionary history. Nitrogen isotopes indicate trophic level, showing exactly where each species fit in the food web hierarchy. Some fossil sites preserve evidence of direct ecological interactions with stunning clarity. The famous fighting dinosaur specimen from Mongolia captures a velociraptor and protoceratops locked in mortal combat, preserved at the moment of their struggle by a sudden sandstorm. This single fossil provides direct evidence of predator-prey relationships, fighting behavior, and the specific hunting techniques employed by these ancient predators. Bite marks on fossil bones often tell stories of scavenging hierarchies and feeding behaviors. Analysis of tooth marks on dinosaur bones has revealed complex patterns of carcass utilization, showing that different predator and scavenger species accessed carcasses in predictable sequences based on their size and jaw strength. Large tyrannosaurs left distinctive deep puncture marks, while smaller theropods left different damage patterns, allowing paleontologists to reconstruct the entire community of carcass processors in ancient ecosystems. The preservation of stomach contents in some exceptional fossils provides the most direct evidence possible of ancient diets and feeding behaviors. Several dinosaur specimens have been found with their last meals preserved in their stomach cavities, including fish bones in the stomach of baryonyx and plant material in various herbivorous species. These discoveries confirm dietary interpretations based on tooth structure, while often revealing unexpected dietary diversity. Plant fossils provide crucial information about the primary productivity that supported ancient animal communities. The evolution of flowering plants during the Cretaceous period fundamentally changed terrestrial ecosystems, providing new food sources and creating more complex habitat structures. Fossil pollen assemblages show the gradual spread of these new plant communities and their coevolution with insect pollinators and herbivorous animals. The relationship between plants and their pollinators is beautifully preserved in some fossil deposits. Amber inclusions occasionally capture ancient insects with pollen grains still attached to their bodies, providing direct evidence of pollination relationships that existed over 100 million years ago. These tiny fossils reveal that complex plant-insect interactions evolved much earlier than previously suspected and helped drive the diversification of both flowering plants and their insect partners. Seed dispersal relationships are also documented in the fossil record through the preservation of seeds in animal coprolites and stomach contents. Some large herbivorous dinosaurs appear to have swallowed stones to aid in digestion, and these gastroliths are often found associated with plant material, suggesting that these animals played important roles in seed dispersal across ancient landscapes. The structure of ancient forests can be reconstructed from fossilized tree trunks, leaves, and root systems preserved in growth position. These fossil forests reveal how plant communities were organized and how they changed through time in response to climate shifts and evolutionary innovations. The famous Gilboa Forest in New York, dating to about 380 million years ago, preserves an entire ancient forest floor complete with tree stumps and root systems, showing how early trees colonized terrestrial environments. Trace fossils provide additional insights into ecological relationships through the preservation of interaction networks. Boring traces in fossil shells show evidence of predator-prey relationships between drilling gastropods and their bivalve prey. The size and frequency of these borings can be used to reconstruct predation pressure and its effects on prey evolution over geological time. Symbiotic relationships are occasionally preserved in the fossil record, though they require exceptional preservation conditions. Some fossil corals preserve evidence of the algae that lived within their tissues, similar to modern coral-algae partnerships. These relationships were crucial for building the massive reef systems that dominated many ancient marine environments and supported diverse communities of associated organisms. The fossil record also documents how ecological relationships changed through time in response to environmental perturbations. Mass extinction events provide natural experiments in ecosystem collapse and recovery, showing how surviving species reorganized into new ecological configurations. The end Cretaceous extinction eliminated non-avian dinosaurs, but created new opportunities for mammals, which rapidly diversified to fill vacant ecological niches. Recovery patterns following mass extinctions reveal fundamental principles about how ecosystems reorganize after major disruptions. Primary producers typically recover first, followed by primary consumers, and finally by top predators. This pattern is clearly documented in the fossil record following several major extinction events and provides insights into ecosystem stability and resilience. The colonization of new environments is also well documented through fossil evidence. The transition from marine to terrestrial environments during the Devonian period involved not just the evolution of new anatomical features, but also the development of entirely new ecological relationships. Early terrestrial ecosystems were much simpler than modern ones, with fewer trophic levels and less complex interaction networks. Island biogeography principles are illustrated through fossil evidence from isolated landmasses and island chains. Madagascar and other large islands preserve fossil evidence of unique evolutionary radiations that occurred in isolation from mainland faunas. These island fossils show how geographic isolation leads to the evolution of endemic species and simplified ecosystem structures compared to continental areas. The fossil record reveals that ecosystem complexity has generally increased through geological time, though this trend has been interrupted repeatedly by mass extinction events. Early marine communities were dominated by simple filter feeders and deposit feeders, while modern marine ecosystems include complex predator-prey networks and sophisticated behavioral interactions. Co-evolutionary relationships between different groups of organisms are documented through coordinated changes in the fossil record. The evolution of grasses during the Cenozoic era was accompanied by corresponding changes in herbivorous mammal teeth and digestive systems, showing how plant and animal evolution proceeded in tandem to create new ecological partnerships. The preservation of ancient ecosystems in exceptional fossil deposits, like the Burgess Shale, reveals community structures that have no modern analogues. These ancient marine communities included body plans and ecological strategies that were later eliminated by extinction, reminding us that modern ecosystems represent only a small fraction of the ecological possibilities that life has explored. Seasonal patterns in ancient ecosystems can sometimes be reconstructed from fossil evidence. Growth rings in fossil corals and mollusk shells record seasonal temperature and productivity cycles, while the timing of reproductive events can be inferred from the seasonal distribution of juvenile fossils in some deposits. Migration patterns of ancient animals are documented through the geographic distribution of fossils and through isotope analysis of teeth and bones. Some dinosaur species show evidence of seasonal migration between different latitudes, similar to modern large herbivores, while marine reptiles show evidence of long-distance movements between feeding and breeding areas. The fossil record provides our best evidence for understanding how ecosystems respond to long-term environmental change. Climate shifts documented through geological evidence are accompanied by predictable changes in fossil assemblages, showing how species distributions and community compositions track environmental gradients through both space and time. Understanding these ancient ecological relationships provides crucial insights for modern conservation biology and ecosystem management. The deep time perspective offered by fossils shows that ecosystems are dynamic systems that constantly change in response to environmental pressures, but also that recovery from major disruptions can take millions of years and often results in fundamentally different community structures. Perhaps most importantly, the ecological relationships preserved in fossils remind us that no organism exists in isolation. Every species is embedded in a complex web of interactions with other organisms and with the physical environment. These relationships shape the evolution of life on Earth and continue to influence the structure and function of modern ecosystems. The study of fossil ecosystems also reveals the importance of keystone species, whose presence or absence dramatically affects entire communities. The extinction of large herbivorous dinosaurs at the end of the Cretaceous period eliminated the primary consumers that maintained open habitats, leading to fundamental changes in vegetation structure and the evolution of new plant communities adapted to closed forest environments. As we examine these ancient ecological relationships, we begin to appreciate that the history of life is not simply a catalogue of individual species, but rather the story of how organisms have interacted with each other and with their changing environments over billions of years. This ecological perspective transforms our understanding of evolution from a process affecting isolated lineages to one involving entire communities and ecosystems. The complexity revealed in ancient ecosystems also highlights the sophistication of life's response to environmental challenges. Even the earliest complex communities show evidence of intricate relationships between species, suggesting that ecological complexity is not a recent innovation, but rather a fundamental property of life that emerged early in evolutionary history and has been refined over geological time. The study of these ancient ecological relationships continues to yield new insights as analytical techniques improve and new fossil deposits are discovered. Each new find adds another piece to the puzzle of how life has organized itself into communities and how these communities have changed through the vast expanse of geological time, providing an ever richer picture of the dynamic nature of life on Earth. Climate leaves its signature in fossils as clearly as it does in ice cores or tree rings, but the record preserved in stone extends much deeper into the past and reveals climate changes on scales that dwarf modern concerns. The distribution of ancient life across the globe provides a detailed map of how Earth's climate has shifted over hundreds of millions of years, showing warm periods when palm trees grew in polar regions and ice ages when glaciers advanced far into what are now temperate zones. Fossil plants serve as particularly sensitive indicators of ancient climate conditions. Different plant families have specific temperature and moisture requirements, and their fossil distributions reveal climate patterns with remarkable precision. The presence of cycads and tree ferns in Antarctica during the Permian period indicates that this now frozen continent once enjoyed a warm, humid climate that could support lush forests. Similarly, the appearance of deciduous trees in the fossil record marks the development of seasonal climate patterns, as these trees evolved to cope with pronounced wet and dry seasons. The size and shape of fossil leaves provide quantitative estimates of ancient temperatures and rainfall. Modern botanical studies have established clear relationships between leaf characteristics and climate variables. Large leaves with smooth edges indicate warm, wet conditions, while small leaves with serrated edges suggest cooler, drier climates. Paleobotanists use these relationships to reconstruct detailed climate maps for ancient time periods, showing how temperature and precipitation patterns have shifted as continents moved and mountain ranges rose. Fossil corals offer another window into ancient climate conditions, particularly ocean temperatures and chemistry. Modern reef corals have strict temperature requirements and cannot survive in water that is too hot or too cold. When paleontologists find extensive coral reef deposits in areas that are now too cold to support corals, they can infer that ancient ocean temperatures were significantly warmer than today. The chemistry preserved in coral skeletons also records ancient seawater conditions, including acidity levels and the concentration of dissolved minerals. Tree rings preserved in fossilized wood reveal seasonal climate patterns stretching back millions of years. Just as modern trees record wet and dry years in the width and structure of their annual growth rings, ancient trees preserved similar records of year-to-year climate variability. Some fossilized trees show evidence of severe droughts or unusually wet periods that affected growth hundreds of millions of years ago, providing insights into natural climate cycles that operated long before human influence. The chemistry of fossil shells and bones preserves detailed records of ancient ocean and atmospheric conditions. Oxygen isotope ratios in marine fossils reflect both water temperature and the global volume of ice, allowing paleontologists to reconstruct sea level changes and ice sheet fluctuations. Carbon isotope ratios reveal information about ancient atmospheric carbon dioxide levels and the productivity of marine and terrestrial ecosystems. Ice volume records preserved in marine fossils show that Earth has experienced numerous ice ages throughout its history, not just the recent Pleistocene glaciations that are most familiar to us. During these ancient ice ages, sea levels dropped by hundreds of feet as water was locked up in massive ice sheets, exposing vast areas of the continental shelves that are now underwater. The fossil record documents how marine life adapted to these dramatic sea level changes and how terrestrial organisms migrated as climate zones shifted. Greenhouse periods in Earth's history are equally well documented through fossil evidence. During the Eocene thermal maximum, about 56 million years ago, global temperatures rose rapidly due to massive releases of carbon dioxide into the atmosphere. Fossil evidence from this period shows that crocodilians lived in the Arctic, palm trees grew in Wyoming, and tropical conditions extended much farther toward the poles than they do today. The fossil record of this ancient warming event provides crucial insights into how ecosystems respond to rapid climate change. The movement of climate zones through geological time is clearly recorded in the shifting distributions of fossil species. Groups of organisms that are now restricted to tropical regions have fossil records that extend far into what are now temperate and even polar areas. During warm periods, tropical species expanded their ranges toward the poles, while during cold periods, they contracted toward the equator. These range shifts are preserved as clear patterns in the geographic distribution of fossils through rock sequences of different ages. Mountain building events have profoundly influenced climate patterns throughout Earth's history, and these effects are recorded in changing fossil assemblages. The rise of the Himalayas during the Cenozoic era altered global atmospheric circulation patterns and contributed to the gradual cooling that led to the ice ages of the Pleistocene. Fossil evidence shows how these climate changes affected vegetation patterns and animal distributions across vast areas of Asia and beyond. Ocean circulation patterns have also changed dramatically through geological time, with major effects on global climate that are recorded in marine fossil assemblages. The opening and closing of ocean gateways as continents moved has repeatedly altered the flow of warm and cold ocean currents, leading to significant climate changes. The closure of the Isthmus of Panama about 3 million years ago changed Atlantic circulation patterns and may have contributed to the intensification of Northern Hemisphere glaciation. Volcanic activity has caused both short-term and long-term climate changes that are clearly recorded in the fossil record. Massive volcanic eruptions can inject so much ash and sulfur compounds into the atmosphere that they cause temporary cooling by blocking sunlight. The fossil record shows evidence of these volcanic winters in the form of temporary disappearances of temperature-sensitive species from certain regions. Long-term volcanic activity, particularly from large igneous provinces, has caused sustained warming periods through the release of massive amounts of carbon dioxide. These volcanic warming events are associated with several mass extinction events in the geological record, showing how extreme climate change can push ecosystems beyond their ability to adapt. The fossil record also reveals how different groups of organisms have responded to climate change throughout Earth's history. Some species adapted by evolving new physiological capabilities. Others migrated to track suitable climate conditions, and still others became extinct when they could not cope with changing environments. These patterns provide crucial insights into which types of organisms are most vulnerable to climate change and which adaptation strategies are most successful. Rapid climate changes in the geological past have generally been associated with increased extinction rates and ecosystem disruption, while gradual changes have allowed more successful adaptation and migration. However, the fossil record shows that even gradual climate changes can have profound effects when they persist for long periods or cross critical thresholds that trigger cascading ecosystem changes. The study of ancient climate changes through fossil evidence has revealed that Earth's climate system has multiple stable states and can shift rapidly between them when certain tipping points are crossed. These insights from deep time provide crucial context for understanding modern climate change and its potential effects on contemporary ecosystems. Perhaps most importantly, the fossil record shows that climate change has been a constant feature of Earth's history and a major driver of evolutionary change. Species that survived multiple climate changes throughout geological time were those that maintained sufficient genetic diversity and ecological flexibility to adapt to changing conditions. This deep time perspective on climate and life provides essential insights for conservation efforts in our current period of rapid environmental change. The precision with which fossils record ancient climate conditions continues to improve as analytical techniques advance. Paleontologists can now extract increasingly detailed climate information from fossil specimens, revealing not just general climate trends but also seasonal patterns, extreme weather events, and even daily temperature cycles in some cases. These detailed climate records from fossils provide the long-term context necessary for understanding the full range of natural climate variability and the rates at which climate systems can change. This information is crucial for distinguishing between natural climate fluctuations and human-induced changes, and for predicting how climate systems might respond to ongoing environmental changes. The story of life's response to climate change over geological time is ultimately one of resilience and adaptation, but also of vulnerability and loss. While life has survived dramatic climate changes in the past, these changes have often come at enormous cost in terms of biodiversity loss and ecosystem reorganization. The fossil record provides both inspiration for life's remarkable adaptability and sobering warnings about the consequences of rapid environmental change. Mass extinction events represent some of the most dramatic chapters in Earth's history, and the fossil record provides our most detailed account of these catastrophic episodes that periodically reshaped the biological world. These events offer natural experiments in ecosystem collapse and recovery, revealing fundamental insights about the resilience and vulnerability of life on our planet. The study of mass extinctions through fossils requires examining patterns of disappearance and survival across multiple rock layers and geographic regions. Paleontologists look for the sudden absence of previously abundant fossil species, changes in diversity patterns, and the emergence of new groups that filled vacant ecological roles. This work requires careful attention to the quality of the fossil record and the completeness of preservation, as apparent extinctions might sometimes reflect gaps in the geological record, rather than actual biological events. The end-Ordovician extinction, which occurred approximately 445 million years ago, provides one of the clearest examples of how climate change can drive mass extinction. This event coincided with a major ice age that dramatically lowered sea levels and altered ocean chemistry. Marine fossils from this period show a clear pattern of extinction among tropical species that were adapted to warm, shallow seas. When ice sheets expanded and sea levels dropped, these warm water habitats largely disappeared, taking their inhabitants with them. The fossil evidence for this extinction is particularly clear in marine sediments, where diverse assemblages of trilobites, brachiopods, and early corals suddenly give way to much simpler communities, dominated by only a few surviving species. The geographic pattern of extinction follows climate zones, with tropical species suffering the heaviest losses, while organisms adapted to cooler conditions showed higher survival rates. The late Devonian extinction, occurring around 375 million years ago, demonstrates how multiple environmental stresses can combine to create extinction conditions. Fossil evidence suggests this extinction unfolded over several million years. With marine life suffering repeated pulses of extinction, the fossil record shows declining diversity in reef communities, with many groups of corals, brachiopods, and other marine organisms disappearing from the rock record. Recent research has linked this extinction to ocean acidification and oxygen depletion, environmental changes that left clear chemical signatures in marine sediments. The fossil evidence shows that organisms with calcium carbonate shells were particularly vulnerable, suggesting that ocean chemistry changes played a crucial role in driving extinctions. This ancient example of ocean acidification provides sobering parallels to current environmental concerns about carbon dioxide absorption by modern oceans. The end Permian extinction, the most severe crisis in life's history, eliminated an estimated 90% of marine species and 70% of terrestrial vertebrate species approximately 252 million years ago. The fossil record shows this extinction was both rapid and global, affecting ecosystems from the equator to the poles and from shallow seas to deep ocean basins. The fossil evidence for this extinction is dramatic and unmistakable. In marine sediments, diverse assemblages of brachiopods, corals, crinoids, and other marine invertebrates suddenly disappear, replaced by thin layers of sediment containing only the simplest surviving organisms. On land, the rich assemblages of synapsid reptiles that dominated Permian ecosystems vanish from the fossil record, replaced initially by much simpler communities dominated by only a few surviving lineages. The environmental conditions that drove this extinction are preserved in the chemical signatures of rocks from this time period. Massive volcanic eruptions in what is now Siberia released enormous quantities of carbon dioxide and sulfur compounds into the atmosphere, causing severe global warming and ocean acidification. The fossil record shows that this environmental crisis persisted for millions of years, preventing normal ecosystem recovery and creating the simplified biological world of the early Triassic period. Recovery from the end Permian extinction took approximately 10 million years, an extraordinarily long time compared to recovery from other mass extinctions. The fossil record shows that early Triassic ecosystems were characterized by low diversity and unusual ecological structures, with some environments dominated by single species that formed vast monocultures. This delayed recovery pattern provides insights into how severe environmental changes can create lasting effects that persist long after the initial crisis has passed. The end Triassic extinction, occurring about 200 million years ago, coincided with the breakup of the supercontinent Pangaea and massive volcanic eruptions that created the Central Atlantic magmatic province. The fossil record shows this extinction particularly affected marine organisms and early dinosaur relatives on land, creating opportunities for the dinosaur lineages that would dominate Mesozoic ecosystems. The marine fossil record from this time shows the disappearance of many groups that had recovered from the end Permian extinction, including various types of marine reptiles and ammonoid cephalopods. On land, the fossil evidence shows the extinction of many early archosaur groups, leaving ecological niches that were subsequently filled by dinosaurs and other surviving lineages. The end Cretaceous extinction, which eliminated non-avian dinosaurs 66 million years ago, provides the most detailed case study of mass extinction processes due to the exceptional quality of the fossil record from this time period. The discovery of the iridium layer at the Cretaceous-Paleogene boundary provided the first clear chemical evidence for an asteroid impact, while subsequent discoveries have revealed a wealth of detail about the extinction process and its immediate aftermath. The fossil evidence from the final days of the Cretaceous period paints a detailed picture of the world that existed just before the extinction. Dinosaur fossils from the very end of the Cretaceous show that these animals were still diverse and apparently thriving right up to the extinction boundary. The sudden disappearance of dinosaur fossils above this boundary, combined with the appearance of impact-related debris, provides compelling evidence for a catastrophic extinction event. Recent discoveries have revealed sites that preserve fossils from the immediate aftermath of the asteroid impact. These deposits contain a mixture of impact debris, including shocked quartz and glass spherules created by the impact, along with fossils of organisms that died during or immediately after the impact event. Some sites preserve evidence of massive tsunamis and wildfires that swept across continents in the hours and days following the impact. The pattern of survival and extinction at the end of the Cretaceous reveals important principles about what determines survival during mass extinction events. Small body size, omnivorous feeding habits, and the ability to burrow or shelter underground all appear to have increased survival chances. Birds survived while non-avian dinosaurs perished. Mammals diversified rapidly after the extinction, and marine organisms that could tolerate environmental stress showed higher survival rates than specialist species. The recovery pattern following the end Cretaceous extinction shows how surviving species rapidly diversified to fill vacant ecological roles. Mammal fossils from the early Paleocene show an explosion of new body sizes and ecological types, as these animals expanded into niches previously occupied by dinosaurs. Plant fossils show similar patterns of rapid diversification, as flowering plants colonized disturbed landscapes. Each mass extinction event in the fossil record shows unique characteristics that reflect the specific environmental conditions that caused it. Climate change, volcanism, asteroid impacts, and ocean chemistry changes have all played roles in different extinction events, and the fossil record preserves evidence of how these various environmental stresses affected different groups of organisms. The study of background extinction rates between major extinction events reveals that species turnover is a constant feature of the fossil record. Even during periods of relative environmental stability, species regularly appear and disappear from the fossil record as evolution continues and environmental conditions gradually change. Mass extinction events represent periods when this normal rate of species turnover accelerated dramatically due to severe environmental stress. The fossil record also preserves evidence of near-extinction events that came close to triggering mass extinctions, but ultimately did not cross that threshold. These events provide insights into the resilience of ecosystems and the factors that determine whether environmental stress leads to localized extinction or global crisis. Smaller-scale extinction events, sometimes called minor extinctions, appear regularly throughout the fossil record and provide opportunities to study extinction processes at more manageable scales. These events often affected particular regions or ecological groups while leaving other parts of the biological world relatively unaffected. The timing of mass extinctions in relation to environmental changes provides crucial insights into cause and effect relationships. In some cases, extinctions appear to follow environmental changes with little delay, suggesting direct causal relationships. In other cases, there may be significant time lags between environmental changes and biological responses, indicating more complex causal mechanisms. The fossil record reveals that some groups of organisms are consistently more vulnerable to extinction than others. Specialist species with narrow ecological requirements tend to suffer higher extinction rates than generalist species with broad environmental tolerances. Large-bodied species are often more vulnerable than small-bodied ones, and species with slow reproductive rates, are more susceptible than those that can reproduce rapidly. Geographic patterns in the fossil record show that extinction events often affected different regions with varying severity. Island biotas and isolated ecosystems frequently suffered higher extinction rates than continental ecosystems, with more diverse species pools and greater opportunities for migration and adaptation. The preservation of extinct ecosystems in the fossil record sometimes allows detailed reconstruction of pre-extinction communities and comparison with post-extinction assemblages. These studies reveal how ecosystem structure and function changed as a result of mass extinction events, providing insights into how biological communities reorganize following major disruptions. Recovery patterns following mass extinctions show consistent features across different events and time periods. Primary producers typically recover first, followed by primary consumers, and finally by higher-level predators. This pattern reflects the fundamental dependence of ecosystem structure on energy flow from plants through successive trophic levels. The timescales of extinction and recovery revealed by the fossil record provide important perspectives on modern biodiversity loss. While the current rate of species extinction appears to rival or exceed that during past mass extinction events, the recovery phase following mass extinctions typically requires millions of years, far longer than human civilization has existed. Innovation and adaptive radiation following mass extinctions have repeatedly led to the evolution of new ecological strategies and body plans. The fossil record shows that these post-extinction radiations often explored evolutionary possibilities that had not been realized during pre-extinction periods, leading to increased biological diversity and complexity over geological time. The study of mass extinctions through fossils has revealed that these events, while destructive, have also played important roles in shaping the history of life on Earth. By eliminating dominant groups and creating ecological opportunities for surviving lineages, mass extinctions have repeatedly redirected the course of evolution and contributed to the overall pattern of increasing biological complexity through geological time. Understanding mass extinctions through the fossil record provides crucial context for evaluating current environmental changes and biodiversity loss. While the specific causes of past and present extinctions may differ, the fundamental principles revealed by fossil evidence about ecosystem vulnerability and resilience remain relevant for conservation efforts and environmental management. The exceptional preservation of extinction boundaries in some fossil sequences allows paleontologists to study these events with remarkable temporal precision. Some sites preserve evidence of changes occurring over periods as short as thousands or even hundreds of years, providing insights into the rapid pace at which ecosystems can collapse under severe environmental stress. The comparison of different mass extinction events through fossil evidence reveals both similarities and differences in how these crises unfolded. While the specific environmental causes varied, the biological responses show consistent patterns that reflect fundamental constraints on how life responds to severe environmental stress. Perhaps most importantly, the fossil record of mass extinctions demonstrates that while life has proven remarkably resilient over geological timescales, this resilience comes at enormous cost in terms of biodiversity loss and ecosystem disruption. The deep time perspective provided by fossils shows that recovery from major environmental crises is possible, but typically requires timescales that dwarf human experience and planning horizons. As we continue to study mass extinctions through fossil evidence, new analytical techniques and discoveries continue to refine our understanding of these crucial events in life's history. Each new piece of evidence adds to our understanding of how ecosystems respond to environmental crisis, and how life has repeatedly rebuilt itself following catastrophic disruption. The lessons learned from studying ancient mass extinctions through fossils provide both hope and warning for our current environmental situation. Life has survived repeated catastrophes and repeatedly diversified to create new forms of biological complexity. However, the cost of these recoveries, measured in lost species and disrupted ecosystems, reminds us of the precious nature of the biological diversity that surrounds us today, the fossil record of mass extinctions ultimately tells a story of both destruction and renewal, showing how life has repeatedly faced existential crises and found ways to survive and diversify. This record provides our best guide for understanding how contemporary ecosystems might respond to ongoing environmental changes and what we might expect from the biological world as it faces the challenges of the Anthropocene epoch. The study of exceptional fossil sites has revolutionized our understanding of prehistoric life by preserving organisms and anatomical details that would normally decay within days or weeks of death. These remarkable deposits, known by the German term Lagerstätten, meaning storage places, provide extraordinary windows into ancient ecosystems by capturing complete communities of organisms, along with their soft tissues, behaviour, and ecological interactions. The Burgess Shale of British Columbia represents perhaps the most famous of these exceptional preservation sites. Dating to approximately 508 million years ago, during the Middle Cambrian period, this deposit has yielded thousands of fossils that preserve not just the hard shells and skeletons that typically fossilize, but also the soft tissues of creatures that would otherwise be completely unknown from the fossil record. The preservation conditions at the Burgess Shale involved rapid burial by fine sediment in an oxygen-free environment at the base of an underwater cliff. The absence of oxygen prevented bacterial decay, while the fine sediment captured microscopic details of anatomy that would normally disappear within hours of death. The result is a fossil assemblage that includes jellyfish, worms, arthropods, and other soft-bodied organisms preserved in exquisite detail. Among the most remarkable Burgess Shale fossils are creatures like Opabenia, with its 5 eyes and bizarre vacuum cleaner-like proboscis, and Hallucigenia, originally reconstructed upside down due to its strange anatomy of defensive spines and tentacle-like legs. These fossils reveal that early animal evolution explored body plans and ecological strategies that have no modern equivalents, showing that the diversity of life forms in ancient oceans was far greater than what typical fossil sites suggest. The Solnhofen limestone of Germany provides another extraordinary example of exceptional preservation from a very different environment. These late Jurassic deposits, approximately 150 million years old, formed in tropical lagoons with extremely salty water that prevented bacterial decay and scavenging. The fine limestone preserves remarkable details of both marine and terrestrial organisms that occasionally fell or were washed into these ancient lagoons. The most famous Solnhofen fossil is undoubtedly Archaeopteryx, the feathered dinosaur that provided crucial evidence for the evolutionary transition between dinosaurs and birds. The preservation of feather impressions in these fossils was revolutionary, showing for the first time that some dinosaurs possessed complex plumage. The detail preserved in Archaeopteryx specimens includes not just the presence of feathers, but their specific arrangement and structure, revealing information about flight capabilities and thermoregulation. Solnhofen has also yielded exceptional fossils of pterosaurs, marine reptiles, and arthropods, all preserved with soft tissue details that provide insights into their biology and behavior. Some specimens preserve stomach contents, showing what these ancient creatures ate, while others show evidence of live birth in marine reptiles, revealing reproductive strategies that would be impossible to infer from skeletal remains alone. The Messel Pit in Germany represents exceptional preservation from a much more recent time period, the Eocene epoch, approximately 47 million years ago. This site formed in an ancient volcanic lake, where periodic releases of toxic gases from the lake bottom killed organisms living in and around the water. The anoxic lake bottom preserved complete skeletons along with soft tissues, stomach contents, and even the outline of fur and feathers in some specimens. Messel fossils include early horses, primates, bats, and birds preserved in remarkable detail. Some specimens show the contents of their last meals, including fruits, insects, and fish, providing direct evidence of ancient diets and feeding behaviors. The preservation of delicate structures like insect wings and bird feathers has revealed evolutionary relationships and ecological adaptations that would be impossible to determine from typical fossil preservation. The preservation of plant material at Messel is equally extraordinary, with leaves showing cellular detail and flowers preserving pollen and other reproductive structures. These plant fossils provide detailed information about ancient forest ecosystems and climate conditions during a time when global temperatures were much warmer than today. The Liaoning province of China has yielded perhaps the most spectacular dinosaur fossils ever discovered, preserving feathered dinosaurs in extraordinary detail. These early Cretaceous deposits, approximately 125 million years old, formed in ancient lake beds where volcanic ash periodically buried entire ecosystems. The fine ash particles captured microscopic details while the rapid burial prevented scavenging and decay. The feathered dinosaur fossils from Liaoning have revolutionized our understanding of dinosaur biology and the origin of birds. Specimens like Sinosauropteryx preserve not just the presence of primitive feathers, but even evidence of their original coloration through the preservation of melanin-containing organelles called melanosomes. These discoveries have shown that feathers evolved long before flight and served various functions including insulation, display, and camouflage. The Liaoning deposits have also preserved early birds, early mammals, and complete forest ecosystems with remarkable fidelity. Some specimens show evidence of brooding behavior in dinosaurs, with adults preserved sitting on nests of eggs in positions identical to modern birds. Others preserve evidence of aerial combat between pterosaurs, frozen in stone at the moment of their struggle. The Ediacara Hills of Australia preserve some of the earliest evidence of complex multicellular life, dating to approximately 550 million years ago. These fossils represent organisms that lived before the evolution of hard skeletons, and their preservation required very specific conditions involving rapid burial by sand in oxygen-free environments. Ediacaran fossils show a variety of body plans that have no clear relationship to modern organisms. Some appear to be early animals, while others may represent evolutionary experiments that left no descendants. The preservation of these soft-bodied organisms provides our only direct evidence of what life was like before the Cambrian explosion of hard-shelled animals. The Rhynie Chert of Scotland preserves an entire early terrestrial ecosystem from the Devonian period, approximately 400 million years ago. This site formed when hot springs deposited silica around living organisms, creating three-dimensional fossils that preserve cellular detail in plants and microscopic animals. The preservation is so exceptional that scientists can study the internal anatomy of 400 million-year-old organisms at the cellular level. Amber deposits represent another type of exceptional preservation, capturing small organisms and organic materials in tree resin that hardened into fossilized amber. These deposits preserve insects, spiders, plant material, and other small organisms in three-dimensional detail, often including soft tissues and even behavior. Some amber inclusions preserve organisms in the act of mating, feeding, or caring for young, providing direct evidence of ancient behaviors. The exceptional preservation found in amber extends to molecular levels, with some specimens preserving ancient DNA and proteins. While claims of very ancient DNA from amber must be viewed with caution due to contamination issues, amber certainly preserves organic materials far better than typical fossilization processes. La Brea tar pits in California provide an example of exceptional preservation from the very recent geological past, preserving Ice Age mammals and birds in asphalt deposits that trapped animals over thousands of years. The preservation in these deposits is so good that scientists can extract ancient DNA and proteins from specimens only tens of thousands of years old. These tar pit fossils provide detailed information about Ice Age ecosystems and the interactions between predators and prey. The large number of carnivore specimens suggests that these were predator traps, where struggling herbivores attracted multiple predators that also became trapped. The study of exceptional preservation sites has revealed that the diversity of ancient life was far greater than what typical fossil sites suggest. Soft-bodied organisms, which make up the majority of modern biodiversity, are virtually absent from normal fossil deposits, but are abundant in Lagerstätten. These sites show that ancient ecosystems included entire categories of organisms that would be completely unknown if we relied only on typical preservation of hard parts. The conditions required for exceptional preservation are rare and specific, which means that Lagerstätten represent only brief snapshots of ancient life rather than continuous records. However, when these snapshots are available, they provide unparalleled insights into the biology, behavior, and ecology of ancient organisms. The technological analysis of exceptionally preserved fossils has revealed information that early paleontologists could never have imagined. Electron microscopy reveals cellular structures in fossils hundreds of millions of years old, while chemical analysis can identify original organic compounds and even reconstruct the colors of ancient organisms. Perhaps most importantly, exceptional preservation sites remind us that the fossil record, while incomplete, occasionally provides remarkably complete information about ancient life. These sites bridge the gap between the limited information available from typical fossils and the rich detail of modern biological studies, allowing paleontologists to study extinct organisms with nearly the same level of detail possible for living species. The discovery and study of New Lagerstätten continues to yield surprising insights about the history of life. Each new site of exceptional preservation has the potential to reveal previously unknown aspects of ancient biology and to challenge existing ideas about how life evolved and functioned in the past. The preservation of behavior and ecological interactions in these exceptional sites provides information that cannot be obtained from any other source. These fossil snapshots capture moments of ancient life in action, showing how extinct organisms lived, moved, fed, and interacted with each other and their environments. As analytical techniques continue to improve, scientists are able to extract increasingly detailed information from exceptionally preserved fossils. These advances promise to reveal even more about the biology and ecology of ancient life, continuing to surprise us with the sophistication and complexity of organisms that lived hundreds of millions of years ago. The study of exceptional preservation ultimately shows us that ancient life was far more diverse, complex, and behaviorally sophisticated than we might expect from typical fossil evidence. These remarkable sites provide our clearest windows into the true nature of prehistoric ecosystems and remind us that the history of life includes chapters of extraordinary richness that are only occasionally preserved in the geological record. Human evolution represents one of the most intensively studied chapters in the fossil record, and the evidence preserved in ancient bones and artifacts tells a detailed story of how our lineage diverged from other primates and gradually acquired the characteristics that define modern humanity. The fossil evidence for human evolution demonstrates both the power of the fossil record to reveal evolutionary relationships and the ongoing nature of scientific discovery as new finds continue to refine our understanding of our own origins. The story of human evolution begins in Africa, with early primates, whose fossils document the gradual development of characteristics that would eventually lead to the human lineage. Fossil sites across eastern and southern Africa have yielded remains spanning millions of years, creating a detailed timeline of human ancestry that continues to grow more complete with each new discovery. The earliest human ancestors, known as Australopithecines, lived between approximately 4 and 2 million years ago and displayed a mixture of ape-like and human-like characteristics. The famous fossil Lucy, discovered in Ethiopia in 1974, belongs to the species Australopithecus afarensis and preserves about 40% of a complete skeleton. Lucy's anatomy shows clear evidence of upright walking, with modifications to the pelvis, leg bones, and spine that enabled bipedal locomotion, while retaining many primitive features in the skull and arms. The preservation of Lucy's skeleton was exceptional for such an ancient specimen, allowing detailed study of her anatomy and locomotion. The shape of her pelvis shows adaptations for walking upright, while the length and curvature of her leg bones indicate an efficient bipedal gait. However, her long arms and curved finger bones suggest that she retained significant climbing abilities, indicating that early human ancestors lived both on the ground and in trees. Footprint evidence provides additional confirmation of early bipedalism in human ancestors. The famous Litoli trackway in Tanzania preserves actual footprints of Australopithecines walking across volcanic ash approximately 3.5 million years ago. These tracks show clear evidence of upright walking, with heel-to-toe gait patterns and foot anatomy similar to modern humans. The preservation of these footprints required precise conditions, with the volcanic ash being just the right consistency to receive clear impressions before being covered and preserved by subsequent ashfalls. The fossil record shows that multiple species of early human ancestors coexisted in Africa, sometimes in the same regions and time periods. Recent discoveries have revealed at least 6 different Australopithecine species, each adapted to slightly different ecological niches. Some species, like Australopithecus robustus, developed massive jaws and teeth for processing tough plant materials, while others, like Australopithecus africanus, maintained more generalized diets. The transition from Australopithecines to the genus Homo is documented by fossils that show gradual increases in brain size and changes in skull shape and dental anatomy. The earliest members of our own genus, such as Homo habilis, lived approximately 2.5 million years ago and are associated with the first stone tools in the archaeological record. Tool use represents a crucial development in human evolution, and the preservation of both tools and the bones they were used to process provides direct evidence of early human behavior. The oldest known stone tools, from sites in Ethiopia, date to approximately 2.5 million years ago and show evidence of systematic manufacture and use. These tools were used to process animal carcasses, as evidenced by cut marks on fossil bones that match the edges of stone implements. The development of increasingly sophisticated tool technologies is clearly documented in the archaeological record. Early tools were simple choppers and scrapers made by removing a few flakes from stone cores. Over time, tool manufacture became more complex, with standardized shapes and evidence of planning and forethought in their production. The preservation of these tools, along with the debris from their manufacture, provides detailed insights into the cognitive development of early humans. Brain size increase is one of the most dramatic trends in human evolution, and this change is clearly documented in fossil skulls spanning the last 3 million years. Australopithecine brains were approximately the same size as modern chimpanzees, while modern human brains are roughly 3 times larger. This increase occurred gradually, with significant acceleration during the last 2 million years of human evolution. The preservation of skull fossils allows detailed study of brain evolution through endocasts, natural or artificial casts of the brain cavity that preserve the shape and size of ancient brains. These studies have revealed not just increases in overall brain size, but also changes in the proportions of different brain regions. Areas associated with language processing and tool use show particular expansion in later human ancestors. The fossil record also documents changes in human anatomy related to diet and lifestyle. Early Australopithecines had large, powerful jaws and teeth adapted for processing tough plant materials. Later, human ancestors show reduction in jaw size and changes in tooth structure that reflect dietary shifts toward higher quality foods, including meat processed with stone tools. The evolution of human locomotion is documented through changes in leg and pelvic anatomy preserved in fossil remains. Early Australopithecines show clear adaptations for upright walking, but retained features suggesting significant climbing ability. Later species show increasingly complete adaptation to terrestrial bipedalism, with longer legs and more efficient walking gaits. The geographic expansion of human ancestors is clearly documented in the fossil record through discoveries spanning multiple continents. The earliest evidence of human ancestors outside Africa comes from the Caucasus region, where fossils at Dmanisi in Georgia preserve remarkably complete skulls and skeletons of early Homo erectus, dating to approximately 1.8 million years ago. The Dmanisi fossils are particularly valuable because they represent an entire population of early humans, including juveniles and adults of different ages. This population sample shows considerable variation in skull size and shape, demonstrating that early human species displayed significant individual differences while sharing fundamental anatomical characteristics. The preservation at this site included not just skulls and teeth, but also postcranial bones that reveal information about body size and locomotion. The fossil evidence shows that these early emigrants from Africa were smaller-brained and more primitive than later human populations, yet they successfully colonized new environments and adapted to different climatic conditions. Their tool assemblages were relatively simple, but they demonstrate the ability to manufacture implements and process food in environments very different from their African homeland. The expansion into Asia is further documented by fossil sites in Java and China that preserve evidence of Homo erectus populations spanning nearly 2 million years. The preservation conditions at some of these sites, particularly in cave deposits, have yielded remarkable insights into early human behavior and adaptation to diverse environments. The Java fossils, first discovered in the 1890s, provided some of the earliest evidence for human evolution outside Africa. These specimens, including the famous Pithecanthropus erectus skull, show brain sizes intermediate between those of Australopithecines and modern humans, documenting the gradual expansion of cognitive capabilities in our lineage. Recent underwater discoveries off the coast of Java have revealed additional human fossils that were submerged when sea levels rose after the last ice age. These finds demonstrate how changes in sea level have hidden important fossil sites beneath modern oceans, suggesting that many crucial pieces of the human evolutionary puzzle remain to be discovered. The Chinese fossil record includes sites like Zhoukudian near Beijing, where extensive deposits have preserved evidence of Homo erectus populations that lived between approximately 700,000 and 200,000 years ago. The preservation at this site included not just human fossils, but also evidence of fire use, tool manufacture, and food processing that provide insights into early human behavior. The use of fire represents a crucial development in human evolution, and the fossil evidence for controlled fire use appears gradually in the archaeological record. Early evidence consists of burned bones and heated sediments that suggest fire use, while later sites preserve clear hearths and evidence of systematic fire management. The development of more sophisticated tool technologies is clearly documented through the archaeological record associated with human fossil sites. The transition from simple choppers to carefully shaped hand axes represents a significant cognitive advance, requiring planning, skill, and the ability to envision the final product before beginning manufacture. The European fossil record documents the later expansion of human ancestors into temperate and even cold climates. Sites like Atapuerca in Spain have preserved evidence of some of the earliest European populations, dating to approximately 1 million years ago. These fossils show adaptations to cooler climates and different food resources than their African and Asian relatives. The Neanderthal lineage, which diverged from our own approximately 600,000 years ago, is extensively documented in European fossil sites. Neanderthal fossils show distinctive anatomical features, including robust skulls, prominent brow ridges, and stocky body builds adapted to cold climates. The preservation of Neanderthal DNA in some fossils has revealed detailed information about their genetics and relationship to modern humans. The fossil evidence shows that Neanderthals were sophisticated toolmakers and hunters who adapted successfully to Ice Age environments across Europe and Western Asia. Their burial practices, evidenced by intentional graves containing stone tools and other artifacts, suggest complex social behaviors and possibly symbolic thinking. The emergence of anatomically modern humans is documented through fossil sites in Africa dating to approximately 300,000 years ago. These early modern human fossils show the characteristic skull shape, reduced brow ridges, and gracile build that distinguish our species from earlier human ancestors. The expansion of modern humans out of Africa and their subsequent colonization of the globe is documented through fossil sites on every continent except Antarctica. The timing and routes of this expansion continue to be refined as new fossil sites are discovered and dated with increasing precision. The interaction between modern humans and other human species, including Neanderthals and the recently discovered Denisovans, is preserved in both fossil and genetic evidence. Some sites preserve evidence of overlapping populations, while ancient DNA studies have revealed that interbreeding occurred between these different human lineages. The development of increasingly sophisticated cultural technologies, including art, music, and complex tool industries, is documented in archaeological sites associated with modern human fossils. The preservation of cave paintings, carved artifacts, and complex tool assemblages shows the rapid development of symbolic behavior and technological innovation. The colonization of Australia approximately 65,000 years ago required sophisticated boat building and navigation skills, demonstrating the advanced cognitive capabilities of early modern humans. The fossil sites in Australia preserve evidence of the unique evolutionary trajectory that human populations followed in isolation on this continent. The settlement of the Americas represents the final chapter in the global expansion of modern humans, with fossil evidence suggesting arrival by at least 15,000 years ago. The preservation conditions in some American sites, including frozen remains and dry cave deposits, have yielded exceptional insights into the lifeways of early American populations. The fossil record of human evolution demonstrates both the gradual nature of evolutionary change and the rapid pace of cultural development in our lineage. While anatomical changes occurred over millions of years, the development of complex technologies and symbolic behaviors accelerated dramatically in the last 100,000 years. The preservation of human fossils has been enhanced by cultural practices, including intentional burial, which increase the likelihood that remains would be protected from scavenging and weathering. This cultural innovation has enriched the later portions of the human fossil record, providing more complete information about recent human populations. The study of human fossils has revealed the importance of Africa as the cradle of human evolution, with all major developments in our lineage first appearing on this continent before spreading to other parts of the world. This pattern is consistent across millions of years of human evolution, from the earliest Australopithecines to the emergence of modern humans. The fossil evidence for human evolution continues to grow as new sites are discovered and excavated around the world. Each new discovery adds detail to our understanding of human origins and helps refine the timeline of our evolutionary development. The integration of fossil evidence with genetic studies of modern human populations has provided unprecedented insights into human evolution and migration patterns. Ancient DNA extracted from fossil remains allows direct comparison between extinct populations and their modern descendants. The preservation of cultural artifacts alongside human fossils provides insights into the development of technology, art, and social organization in our ancestors. These archaeological materials complement the biological evidence preserved in fossil bones and teeth. The study of human evolution through fossils demonstrates the power of the fossil record to document evolutionary relationships and reveal the history of our own species. While gaps remain in our understanding, the overall picture of human evolution is remarkably well supported by fossil evidence spanning millions of years. The fossil record shows that human evolution was not a linear progression toward modern humans, but rather a complex branching pattern with multiple species coexisting and competing for resources. Only one lineage ultimately survived to give rise to modern humanity, while others became extinct without leaving descendants. The environmental context preserved alongside human fossils reveals how climate change and habitat shifts influenced human evolution and migration patterns. Periods of environmental stress often coincided with evolutionary innovations and population movements that shaped the course of human development. The exceptional preservation at some human fossil sites has yielded insights that would be impossible to obtain from typical preservation conditions. Sites with preserved soft tissues, coprolites, and organic artifacts provide intimate details about the lives and behaviors of our ancient ancestors. The ongoing study of human fossils continues to reveal new aspects of our evolutionary history, challenging previous assumptions and adding complexity to our understanding of how modern humans develop their unique characteristics. Each new discovery contributes to the ever-growing body of evidence that documents our remarkable evolutionary journey from African apes to global colonizers. The techniques used to study human fossils have evolved dramatically, allowing scientists to extract increasing amounts of information from precious specimens. Advanced imaging, chemical analysis, and DNA sequencing have revolutionized the field of human paleontology and continue to yield surprising discoveries about our ancient relatives. The fossil record of human evolution stands as one of the most complete and well-documented examples of evolutionary change over extended time periods. This record provides crucial evidence for understanding not just human origins, but also the broader processes of evolution, adaptation, and species change that have shaped life throughout Earth's history. The story revealed by human fossils is ultimately one of remarkable adaptation and innovation, showing how our ancestors successfully colonized diverse environments around the globe while developing the cultural and technological capabilities that distinguish modern humanity. This fossil evidence connects us directly to our ancient past and provides the foundation for understanding our place in the broader history of life on Earth. Recent advances in paleontological techniques have transformed our ability to extract information from fossils, revealing details about prehistoric life that earlier generations of scientists could never have imagined. These new methods combine traditional fieldwork with cutting-edge laboratory analyses, allowing researchers to study ancient organisms with unprecedented precision and scope. High-resolution computed tomography scanning has revolutionized the study of fossil anatomy by allowing scientists to examine internal structures without damaging precious specimens. This technique can reveal brain cavities, inner ear structures, and delicate anatomical features that would be impossible to study through traditional preparation methods. CT scanning has been particularly valuable for studying fossil skulls, where the preservation of internal features provides insights into sensory capabilities, intelligence, and behavior. The application of CT scanning to dinosaur eggs has revealed embryonic development stages and growth patterns that show how these ancient reptiles developed from egg to adult. Some scans have captured embryos in various stages of development, providing insights into dinosaur reproduction and parental care behaviors that would be impossible to infer from adult skeletons alone. Synchrotron radiation facilities, originally developed for physics research, now provide paleontologists with powerful tools for examining fossil microstructures. These intense X-ray beams can reveal cellular details, mineral composition, and organic compounds preserved in fossils with extraordinary resolution. Synchrotron studies have confirmed the preservation of actual cellular structures in fossils hundreds of millions of years old, blurring the line between paleontology and cell biology. Chemical analysis techniques have opened entirely new avenues for studying fossil preservation and ancient biology. Mass spectrometry can identify organic compounds preserved in fossils, revealing information about original biochemistry and preservation processes. Some studies have identified amino acid sequences from ancient proteins, providing direct chemical evidence of evolutionary relationships between extinct and living organisms. Isotope analysis has become a standard tool for studying ancient diets, migration patterns, and environmental conditions. The ratios of different isotopes in fossil bones and teeth reflect the chemistry of ancient food webs and environments. Carbon isotopes reveal whether animals fed on land plants or marine organisms, while oxygen isotopes indicate climate conditions and seasonal patterns. The study of trace elements in fossil bones can reveal information about ancient physiology and metabolism. Elements like strontium and barium concentrations indicate dietary patterns and habitat use, while rare earth elements provide information about preservation conditions and post-mortem alteration of fossil remains. Electron microscopy techniques allow examination of fossil preservation at the molecular level, revealing the processes by which organic materials are replaced by minerals during fossilization. These studies have shown that exceptional preservation sometimes maintains original organic molecules alongside mineral replacements, creating fossils that preserve both morphological and chemical information. Three-dimensional printing technology has transformed the study and display of fossil specimens. Accurate replicas can be produced from CT scan data, allowing researchers to study rare fossils without risking damage to original specimens. These replicas also make important fossils accessible to researchers worldwide and improve public education through museum displays. Digital reconstruction techniques can restore damaged or incomplete fossils, allowing scientists to study anatomy that would otherwise be impossible to interpret. Computer models can test functional hypotheses about extinct organisms, such as jaw mechanics in ancient predators or flight capabilities in early birds. Advanced microscopy techniques have revealed that some fossils preserve original organic materials at the molecular level. The discovery of preserved proteins, DNA fragments, and even cellular organelles in fossils challenges previous assumptions about the limits of preservation and opens new possibilities for studying ancient biology. Laser ablation techniques allow precise sampling of microscopic areas within fossils for chemical analysis without significant damage to specimens. This approach enables detailed studies of growth patterns, seasonal variations, and environmental changes recorded in fossil tissues. The integration of multiple analytical approaches provides comprehensive pictures of ancient organisms and their environments. Combining morphological, chemical, and isotopic data creates detailed reconstructions of how extinct species lived, what they ate, and how they interacted with their environments. Biomolecular paleontology has emerged as a distinct field combining traditional paleontology with molecular biology techniques. The recovery of ancient proteins and DNA from fossils provides direct evidence of evolutionary relationships and reveals aspects of ancient physiology that cannot be inferred from morphology alone. The development of new preparation techniques has improved the quality of fossil specimens available for study. Acid preparation methods can dissolve surrounding rock while preserving delicate fossil structures, revealing anatomical details that would be destroyed by traditional mechanical preparation. Computer modeling and simulation techniques allow scientists to test hypotheses about extinct organisms by creating virtual reconstructions of their anatomy and behavior. These models can simulate locomotion, feeding mechanics, and other aspects of ancient biology that cannot be directly observed in fossils. The application of medical imaging techniques to paleontology has revealed internal structures in fossils that were previously inaccessible. MRI scanning can detect subtle differences in fossil preservation that reveal anatomical features invisible to other techniques. Geochemical analysis of fossil-bearing rocks provides detailed information about ancient environments and preservation conditions. Understanding the chemical environment in which fossils formed helps paleontologists interpret what the fossils represent and how they relate to ancient ecosystems. The use of stable isotope analysis has revealed migration patterns in ancient animals by tracking changes in isotope signatures that reflect different geographic regions and food sources. This technique has been particularly valuable for studying dinosaur migration and the movement patterns of ancient mammals. Advanced dating techniques continue to refine the temporal framework for understanding fossil sequences. New radiometric methods and improved calibration of existing techniques provide increasingly precise ages for fossil sites, allowing detailed correlation of events across different regions and environments. The development of portable analytical equipment allows some analyses to be performed in the field, reducing the need to transport fragile specimens and enabling real-time decision-making during excavations. Portable X-ray fluorescence devices can determine elemental composition of fossils and surrounding rocks on site. Virtual reality and augmented reality technologies are beginning to transform how fossils are studied and displayed. These tools allow researchers to examine three-dimensional fossil reconstructions from multiple angles and to visualize ancient environments and behaviors in immersive ways. The integration of artificial intelligence and machine learning with paleontological research is opening new possibilities for pattern recognition and data analysis. These tools can identify subtle features in large datasets of fossil images and help recognize patterns that might be missed by traditional analysis methods. Remote sensing techniques, including satellite imagery and aerial photography, help paleontologists identify potential fossil sites and plan excavation strategies. These tools can reveal geological formations and sedimentary structures that indicate favorable preservation conditions. The collaboration between paleontologists and engineers has led to new tools for fossil excavation and preparation. Specialized equipment designed for delicate fossil recovery reduces damage during collection and improves the quality of specimens available for study. Database technologies and digital archiving systems are preserving detailed information about fossil specimens and making this data available to researchers worldwide. These digital collections ensure that important scientific information is preserved and accessible for future research. The combination of traditional fieldwork with advanced laboratory techniques continues to yield surprising discoveries about prehistoric life. Each new analytical method opens possibilities for extracting additional information from existing fossil collections, while also improving the study of newly discovered specimens. The ongoing development of new analytical techniques promises to further expand our ability to study fossil preservation and ancient biology. As these methods become more sophisticated and accessible, they will undoubtedly reveal additional secrets about the history of life preserved in the fossil record. The application of these advanced techniques has already transformed our understanding of fossil preservation and ancient biology, showing that fossils contain far more information than previously thought possible. This technological revolution in paleontology continues to surprise scientists with the wealth of data that can be extracted from properly preserved ancient remains. The future of paleontological research will likely see continued integration of multiple analytical approaches, combining traditional morphological studies with advanced chemical, molecular, and digital techniques. This comprehensive approach promises to provide increasingly detailed and accurate reconstructions of prehistoric life and ancient environments. The technological advances in paleontology demonstrate how scientific progress depends on the development of new tools and methods for studying natural phenomena. The fossil record, already rich in information, becomes even more valuable as our ability to extract and interpret that information continues to improve through technological innovation. These analytical advances have also highlighted the importance of proper collection and curation of fossil specimens. As new techniques become available, previously studied fossils often yield additional information, making museum collections invaluable resources for ongoing research and discovery. The democratization of some analytical techniques through improved accessibility and reduced costs has allowed more researchers to apply advanced methods to fossil studies. This broader application of sophisticated techniques has accelerated the pace of discovery and expanded our understanding of prehistoric life across many different groups of organisms and time periods. The integration of advanced analytical techniques with traditional paleontological methods represents a new era in the study of ancient life. This combination of approaches provides the most complete possible picture of prehistoric organisms and their environments, revealing the extraordinary complexity and sophistication of life throughout Earth's history. Despite the remarkable advances in paleontological techniques and the wealth of information preserved in fossils, the fossil record has inherent limitations that must be carefully considered when interpreting evidence about prehistoric life. Understanding these limitations is crucial for drawing accurate conclusions about ancient organisms and avoiding overinterpretation of fossil evidence. The most fundamental limitation of the fossil record is its incompleteness. Paleontologists estimate that the vast majority of species that have ever lived left no fossil trace whatsoever. The process of fossilization requires such specific conditions that most organisms decay completely without preservation. This means that our view of ancient biodiversity is necessarily biased toward organisms and environments that favor preservation. The preservation bias in the fossil record strongly favors organisms with hard parts over those with only soft tissues. Marine organisms with shells, bones, and other mineralizable structures are far more likely to be preserved than soft-bodied creatures like jellyfish, worms, and many arthropods. This bias means that ancient ecosystems dominated by soft-bodied organisms are poorly represented in the fossil record, even though they may have been the most diverse components of ancient communities. Terrestrial environments are generally less favorable for fossilization than marine settings, creating a geographic bias in the fossil record. Upland areas subject to erosion are particularly unlikely to preserve fossils, while lowland areas with active sedimentation provide much better preservation potential. This bias means that highland ecosystems and the organisms adapted to them are underrepresented compared to lowland and marine communities. The temporal distribution of the fossil record is also uneven, with more recent time periods generally better represented than older ones. This pattern reflects both the greater volume of younger sedimentary rocks and the effects of geological processes that can destroy or alter older rock formations. Very ancient fossils require exceptional preservation conditions and are correspondingly rare. The size bias in fossil preservation favors larger organisms over smaller ones. Large bones and shells are more likely to survive the various processes that can destroy organic remains, while small organisms and juvenile specimens are often destroyed before fossilization can occur. This bias can create misleading impressions about the age structure and body size distributions of ancient populations. Behavioral information is only rarely preserved in the fossil record, limiting our understanding of how extinct organisms actually lived and interacted. While trace fossils provide some behavioral insights, most aspects of ancient animal behavior must be inferred from anatomical evidence and comparisons with modern organisms. This indirect approach can lead to incorrect assumptions about ancient lifestyles and capabilities. The fossil record provides limited information about soft tissues and organ systems that rarely preserve. Details about ancient nervous systems, digestive organs, and reproductive anatomy are usually absent from fossil evidence, forcing paleontologists to infer these characteristics from skeletal anatomy and modern analogues. Ecological relationships between ancient organisms are often difficult to reconstruct from fossil evidence alone, while some direct interactions are preserved as bite marks or trace fossils. Most ecological connections must be inferred from the co-occurrence of fossils and comparison with modern ecosystems. These inferences can be incorrect if ancient ecological relationships differed significantly from modern patterns. The chemical and molecular information preserved in fossils is often altered by diagenetic processes that occur after burial. While some original organic compounds can survive for millions of years, they are often modified or replaced by minerals, making it difficult to determine original biochemistry and physiology from fossil evidence. Dating fossils accurately can be challenging, particularly for specimens that lack clear stratigraphic context or are found in complex geological settings. Reworked fossils, where older specimens are eroded and redeposited in younger sediments, can create misleading age relationships. Contamination of specimens with younger organic material can also affect radiometric dating results. The geographic distribution of fossil sites is uneven, with some regions and time periods much better sampled than others. Political, economic, and logistical factors affect where paleontological research is conducted, creating sampling biases that may not reflect the true global distribution of ancient life. The preservation of transitional forms in the fossil record is particularly challenging because these organisms often existed for relatively brief periods during times of rapid evolutionary change. The rarity of transitional fossils can create apparent gaps in evolutionary sequences, even when evolution proceeded gradually. Environmental conditions during fossilization can affect preservation quality and the types of information that survive. Acidic conditions can dissolve shells and bones, while alkaline conditions may favor preservation but alter original chemistry. Temperature, pressure, and groundwater chemistry all influence what aspects of ancient organisms are preserved. The interpretation of fossil evidence requires assumptions about how extinct organisms functioned and lived. These interpretations are often based on studies of modern organisms, but ancient species may have had unique adaptations and behaviors that have no modern parallels. This limitation can lead to incorrect reconstructions of ancient biology and ecology. Taphonomic processes that affect fossils between death and final burial can significantly alter the information preserved in fossil assemblages. Scavenging, transport, and selective preservation can create fossil accumulations that do not accurately represent the original living communities. The resolution of the fossil record varies greatly, with some deposits preserving snapshots of brief moments, while others represent time-averaged accumulations spanning thousands or millions of years. This temporal mixing can obscure short-term ecological patterns and evolutionary events. Pathological specimens, while providing insights into ancient diseases and injuries, represent unusual conditions that may not be typical of normal ancient populations. Care must be taken not to generalize from pathological fossils to entire species or populations. The fossil record cannot preserve many aspects of ancient physiology and metabolism that are crucial for understanding how extinct organisms functioned. Body temperature, metabolic rate, and physiological adaptations must often be inferred from indirect evidence, leading to ongoing debates about fundamental aspects of ancient biology. Behavioral complexity and intelligence are particularly difficult to assess from fossil evidence. While brain size and structure can be estimated from skull fossils, the relationship between brain anatomy and cognitive capabilities is complex and may have been different in extinct organisms than in modern animals. The preservation of juvenile stages and reproductive biology is often poor in the fossil record, limiting understanding of ancient life cycles and developmental patterns. Eggs, embryos, and juvenile specimens require exceptional preservation conditions and are correspondingly rare in fossil assemblages. Seasonal and annual variations in ancient ecosystems are usually lost in the fossil record, which typically represents time-averaged accumulations. This limitation makes it difficult to understand how ancient communities responded to environmental cycles and short-term climate variability. The co-occurrence of fossils in the same rock layer does not necessarily mean that the organisms lived together or interacted ecologically. Time averaging and transport can bring together fossils of organisms that lived at different times or in different habitats. Despite these significant limitations, the fossil record remains our most direct source of information about the history of life on Earth. Recognizing and accounting for these biases and limitations allows paleontologists to extract reliable information about prehistoric life while avoiding overinterpretation of incomplete evidence. The key to successfully using fossil evidence lies in understanding what fossils can and cannot tell us, and in combining multiple lines of evidence to build comprehensive pictures of ancient life. When interpreted carefully and with full awareness of their limitations, fossils provide remarkable insights into the history, diversity, and evolution of life on our planet. The ongoing discovery of new fossil sites, improvement in analytical techniques, and better understanding of preservation processes continue to expand what can be learned from fossil evidence. While the limitations of the fossil record will never be completely overcome, continued research steadily improves our ability to extract accurate information about prehistoric life from the evidence preserved in stone. Modern paleontological research increasingly emphasizes the importance of understanding taphonomic processes and preservation biases when interpreting fossil assemblages. This approach helps ensure that conclusions about ancient life are based on solid evidence rather than artifacts of preservation or collection biases. The integration of fossil evidence with other sources of information, including molecular phylogenies, biogeographic patterns, and comparative studies of modern organisms, helps overcome some limitations of the fossil record by providing independent tests of paleontological interpretations. Ultimately, the limitations of the fossil record remind us that our understanding of prehistoric life is necessarily incomplete and subject to revision as new evidence becomes available. This uncertainty is not a weakness of paleontology, but rather a reflection of the scientific method's emphasis on hypothesis testing and the continuous refinement of knowledge based on new observations and evidence. The study of fossil preservation and taphonomy has become a sophisticated field in its own right, helping paleontologists understand how the fossil record formed and how to interpret it more accurately. This knowledge improves our ability to distinguish between genuine biological patterns and artifacts of preservation, leading to more reliable reconstructions of ancient life. As we reach the end of our gentle journey through deep time, it becomes clear that fossils offer us something truly extraordinary, a direct connection to worlds that vanished long before human civilization began. Each specimen we've explored tonight, from the humblest trace of an ancient burrow to the most spectacular preserved dinosaur skeleton, represents a minor miracle of preservation that allows us to touch, quite literally, the deep history of our planet. The story written in stone is one of constant change and remarkable resilience. Over billions of years, life has faced countless challenges, from devastating asteroid impacts to gradual climate shifts, from the rise and fall of mountain ranges to the opening and closing of ocean basins. Through it all, organisms have adapted, evolved, and diversified, creating the rich tapestry of life that surrounds us today. What makes this story so compelling is its foundation in tangible evidence. Unlike myths or speculation, every detail we've discussed is grounded in actual specimens that can be examined, measured, and studied. The footprints of dinosaurs pressed into ancient mud, the delicate impressions of feathers preserved in limestone, the chemical signatures locked in fossilized shells, all provide direct testimony about how life has changed through time. The techniques we've explored for studying these ancient remains demonstrate the remarkable ingenuity of modern science. From CT scanners that peer inside fossil skulls to mass spectrometers that identify organic compounds millions of years old, technology has transformed our ability to extract information from these stone archives. Yet, at its heart, paleontology remains a deeply human endeavor, driven by curiosity about the ancient world and the patient work of countless researchers who have dedicated their lives to reading the messages written in stone. Perhaps most remarkable of all is how this evidence challenges and refines our understanding of life's history. The discovery of feathered dinosaurs transformed our image of these ancient reptiles from sluggish, cold-blooded monsters into active, possibly warm-blooded creatures with complex behaviors. The recognition of mass extinction events revealed that life's history has been punctuated by crises that reshaped entire ecosystems. The documentation of gradual climate changes showed how life responds to environmental shifts over geological timescales. These discoveries remind us that science is not a collection of fixed facts, but rather an ongoing conversation between evidence and interpretation. Each new fossil find has the potential to revise our understanding of prehistoric life, keeping paleontology a vibrant and dynamic field where surprises await around every rocky outcrop and beneath every layer of sediment. The fossil record also provides crucial perspective on our current environmental situation. The deep time view revealed through fossils shows that while life has survived dramatic changes in the past, these changes often came at enormous cost in terms of biodiversity loss and ecosystem disruption. The recovery from mass extinctions typically required millions of years, timescales that dwarf human experience and planning horizons. Yet fossils also demonstrate life's remarkable creativity and resilience. After every major crisis documented in the fossil record, surviving organisms have diversified into new ecological roles and developed innovative solutions to environmental challenges. The mammals that inherited the world after the dinosaurs' extinction, the flowering plants that transformed terrestrial ecosystems, the countless species that adapted to changing climates and shifting continents, all testify to life's extraordinary ability to reinvent itself. The study of fossils has revealed that evolution is neither random nor predictable, but rather a complex interplay between chance events and natural selection. Environmental changes create new opportunities and challenges, while the existing diversity of life provides the raw material for evolutionary innovation. The result is a history full of unexpected twists and remarkable adaptations that no human imagination could have conceived. Understanding this deep history of life also provides perspective on our own place in the natural world. Humans represent just the latest chapter in a story that began billions of years ago, and our species has existed for only a tiny fraction of life's long journey. Yet in that brief time, we have developed the remarkable ability to read and interpret the fossil record, allowing us to understand our own origins and the history of the planet we inhabit. The technological advances that have transformed paleontology continue to reveal new secrets hidden in ancient rocks. Molecular techniques are pushing back the boundaries of what organic materials can survive over geological time, while imaging technologies are revealing internal structures and microscopic details that were previously invisible. Each new analytical method opens possibilities for extracting additional information from fossil specimens, ensuring that the study of prehistoric life will continue to yield surprises for generations to come. The democratization of some paleontological techniques through improved technology and reduced costs has also expanded the community of researchers studying fossils. Citizen scientists, students, and researchers in developing countries now have access to analytical tools that were once available only at major research institutions, broadening the global effort to understand Earth's biological heritage. Museum collections around the world preserve millions of fossil specimens, many of which have never been studied in detail. As new techniques become available and research priorities shift, these collections represent vast repositories of untapped scientific information. Specimens collected decades or even centuries ago often yield new insights when examined with modern methods, making museum curation and preservation efforts crucial for ongoing research. The educational value of fossils extends far beyond professional research. These tangible remnants of ancient life capture public imagination in ways that few other scientific subjects can match. Children and adults alike are fascinated by dinosaurs, ancient marine reptiles, and other prehistoric creatures, making fossils powerful tools for teaching about evolution, deep time, and the scientific method. The preservation of fossil sites and the protection of fossil resources represent important conservation challenges for the modern world. Many of the most productive fossil localities are threatened by development, mining, and other human activities. Ensuring that future generations have access to these windows into the past requires careful management and protection of fossil-bearing geological formations. International cooperation in paleontological research has become increasingly important as scientists recognize that understanding life's history requires a global perspective. Fossil evidence from different continents and environments must be integrated to reconstruct ancient biogeographic patterns and understand how life responded to planetary-scale environmental changes. The training of new paleontologists combines traditional fieldwork skills with increasingly sophisticated laboratory techniques. Modern paleontologists must be competent in everything from careful excavation and specimen preparation to advanced chemical analysis and computer modeling. This broad skill set reflects the interdisciplinary nature of contemporary paleontological research. The communication of paleontological discoveries to the public represents both an opportunity and a responsibility for the scientific community. Accurate reporting of fossil discoveries helps build public understanding and support for scientific research, while sensationalized or inaccurate coverage can spread misconceptions about prehistoric life and the scientific process. The fossil record continues to surprise scientists with evidence of ancient complexity and sophistication. Recent discoveries have revealed that many behaviors and capabilities once thought to be uniquely modern actually have deep evolutionary roots. Complex social behaviors, sophisticated reproductive strategies, and even tool use appear much earlier in the fossil record than previously suspected. The study of exceptional preservation sites continues to yield remarkable insights about ancient soft tissue anatomy and behavior. These rare windows into complete ancient ecosystems show that the diversity of prehistoric life was far greater than what typical fossil sites suggest, reminding us that most of life's history remains hidden from view. The integration of fossil evidence with molecular phylogenies and developmental biology has created new synthetic approaches to understanding evolutionary history. These interdisciplinary studies reveal how genetic changes translate into morphological evolution and how developmental constraints influence the paths that evolution can take. Climate change research increasingly relies on fossil evidence to understand how ecosystems respond to environmental perturbations over long timescales. The detailed climate records preserved in fossils provide crucial baseline data for understanding natural variability and predicting future changes in Earth's environmental systems. The discovery of life in extreme environments on Earth has influenced how paleontologists interpret ancient ecosystems and search for evidence of early life. Organisms living in conditions once thought impossible for life have expanded our understanding of where and how life can survive, leading to new interpretations of ancient environments and preservation conditions. The application of engineering principles to paleontology has revealed new insights about how extinct organisms functioned. Biomechanical analyses of fossil bones and shells can determine how ancient animals moved, fed, and interacted with their environments, providing detailed reconstructions of ancient behaviors and capabilities. The study of co-evolutionary relationships through the fossil record has shown how different groups of organisms have influenced each other's evolution over geological time. Plant-animal interactions, predator-prey relationships, and symbiotic associations all leave traces in the fossil record that reveal the complex networks of relationships that shaped ancient ecosystems. The documentation of evolutionary innovations through fossil evidence provides insights into how major transitions in life's history occurred. The evolution of photosynthesis, multicellularity, hard skeletons, flight, and other key innovations can be traced through fossil sequences that show how these capabilities developed over time. The fossil record of microbial life, while challenging to study due to preservation limitations, provides our only direct evidence of life's earliest chapters. These ancient microfossils document the emergence of cellular life and the gradual development of the complex biochemical processes that support all modern organisms. The preservation of ancient environments in fossil assemblages allows reconstruction of ecological communities that have no modern analogues. These extinct ecosystems reveal alternative ways that life can organize itself and provide insights into the range of ecological possibilities that evolution has explored. The study of fossil preservation processes continues to reveal new mechanisms by which organic materials can survive over geological time. Understanding these processes helps paleontologists recognize and interpret different types of preservation, while also suggesting new places to search for exceptionally preserved specimens. The application of advanced imaging techniques to fossil studies has revealed internal structures and microscopic details that transform our understanding of ancient anatomy. These techniques allow non-destructive examination of rare specimens while providing unprecedented resolution of fossil preservation. The development of new analytical methods for studying fossil chemistry has opened possibilities for extracting information about ancient metabolism, physiology, and even molecular biology. These techniques push the boundaries of what can be learned from fossil specimens, while providing new tools for testing hypotheses about ancient life. The integration of multiple dating techniques has improved the temporal framework for understanding fossil sequences and evolutionary events. Precise ages for fossil sites allow detailed correlation of biological and environmental changes across different regions and time periods. The recognition of taphonomic biases and preservation limitations has led to more sophisticated approaches to interpreting fossil assemblages. Understanding what fossils can and cannot tell us helps ensure that paleontological interpretations are based on solid evidence rather than artifacts of preservation or sampling. The expansion of paleontological research into new geographic regions continues to reveal previously unknown aspects of life's history. Fossil discoveries from underexplored areas often challenge existing ideas about biogeographic patterns and evolutionary relationships. The study of fossil ecosystems provides insights into how biological communities respond to environmental change over different timescales. These deep time perspectives on ecosystem dynamics inform modern conservation efforts and help predict how contemporary ecosystems might respond to ongoing environmental changes. The documentation of evolutionary rates through fossil evidence shows that the pace of change has varied dramatically throughout life's history. Some lineages remained essentially unchanged for hundreds of millions of years, while others underwent rapid morphological evolution during relatively brief intervals. The fossil record of extinction and recovery reveals fundamental principles about ecosystem resilience and the factors that determine survival during environmental crises. These insights have direct relevance for understanding and managing contemporary biodiversity loss. The preservation of reproductive structures and developmental stages in fossils provides insights into the evolution of life cycles and reproductive strategies. These discoveries reveal how fundamental biological processes have changed over geological time and how they have influenced evolutionary patterns. The study of trace fossils continues to yield new insights about ancient behaviors and ecological interactions. These behavioral fossils often preserve information that cannot be obtained from body fossils alone, providing unique windows into how extinct organisms lived and interacted with their environments. The application of statistical methods to large fossil data sets has revealed patterns in the history of life that were not apparent from smaller-scale studies. These macroevolutionary analyses provide insights into the large-scale processes that have shaped biodiversity over geological time. The integration of experimental approaches with fossil studies has provided new insights into preservation processes and ancient biology. Laboratory experiments can test hypotheses about how fossils formed and how ancient organisms functioned, complementing observational studies of fossil specimens. The development of virtual paleontology through digital modeling and simulation has created new possibilities for studying and sharing fossil discoveries. These technologies allow detailed analysis of fossil specimens while making important discoveries accessible to researchers and the public worldwide. The collaborative nature of modern paleontological research brings together specialists from many different fields to address complex questions about ancient life. This interdisciplinary approach combines expertise in geology, biology, chemistry, physics, and computer science to extract maximum information from fossil evidence. The ongoing discovery of new fossil sites around the world ensures that our understanding of prehistoric life will continue to grow and evolve. Each new locality has the potential to preserve unique aspects of ancient ecosystems and reveal previously unknown chapters in life's long history. The importance of long-term research programs in paleontology cannot be overstated. Many of the most significant discoveries have emerged from decades of systematic work at particular sites or on specific groups of organisms. This sustained effort allows detailed documentation of evolutionary patterns and environmental changes, the training of local researchers, and the development of paleontological expertise in countries with rich fossil resources has become increasingly important for global understanding of life's history. These efforts ensure that fossil discoveries can be properly studied and preserved regardless of where they occur. The ethical considerations surrounding fossil collection and trade have become increasingly important as the commercial value of fossils has grown. Ensuring that scientifically important specimens are preserved for research while respecting the rights of local communities and landowners requires careful consideration of legal and ethical frameworks. The role of amateur fossil collectors and citizen scientists in paleontological discovery continues to be significant. Many important fossils are discovered by enthusiasts who then work with professional paleontologists to ensure proper study and preservation of their finds. Through this gentle exploration of deep time, we've discovered that fossils are far more than curiosities or museum displays. They are Earth's memory, preserving the most remarkable story ever told in pages of stone and layers of sediment. Every specimen we've encountered tonight, from the delicate impression of an ancient leaf to the massive bones of long-extinct giants, represents a moment when time itself paused to capture life in its endless dance of change and adaptation. The evidence speaks with quiet authority about worlds that existed long before our species drew its first breath. These ancient testimonies, written in mineral and chemistry, in footprint and bone, remind us that we are part of an unbroken chain of life stretching back billions of years. The same forces that shaped our earliest ancestors continue to work today, connecting us intimately to every creature that has ever lived on this remarkable planet. As you drift toward rest tonight, perhaps you'll carry with you the gentle wonder of knowing that beneath our feet lie the stories of countless generations, patiently waiting to share their secrets with those curious enough to listen to the whispered messages preserved in stone.